Staphylococcus aureus, sometimes just called Staph aureus, is cockle or round shape and grows in clusters. In fact, its name, broken down, means golden cluster of grapes. It sort of starts making sense if you look at it under a microscope. It tends to grow in sticky clusters, and it stains purple when gram-stained due to its peptidoglycan cell wall. So it's gram-positive and it resembles grapes. As for its golden color, when it's grown on blood agar plates, the colonies have a distinctive golden yellow color. Staphylococcus aureus are gram-positive and facultative anaerobes, meaning that they can survive in aerobic and anaerobic environments. They're non-motile and don't form spores. Staphylococci produce an enzyme called catalase, which converts hydrogen peroxide to water and oxygen. Other common cocci, such as streptococci and enterococci, are catalase negative, so they don't have this ability, and we can use a few drops of hydrogen peroxide to differentiate them. Catalase positive bacteria will foam up, while with catalase negative bacteria, nothing happens. Now, a couple of other Staphylococci species, like Staph epidermidis and Staph saprophyticus, are also catalase positive. So to distinguish between them, we can look for another enzyme that's made by Staph aureus, called coagulase. Coagulase converts fibrinogen into fibrin. So let's say that we stir up some Staph aureus bacteria in a liquid emulsion, and then add a few drops of plasma which contains fibrinogen. The coagulase positive Staph aureus will convert the soluble fibrinogen to sticky fibrin, which then visibly clumps up, whereas coagulase negative bacteria won't. Staph aureus is extremely common, and about a quarter of the population is colonized by it, usually in their nostrils, groin, armpits, and other parts of their skin. But most of the time, it's a normal part of our skin flora, and doesn't cause trouble. The skin flora is a complex ecosystem of different bacterial species, and occasionally Staph aureus can start to dominate that ecosystem. In individuals that have Staph aureus colonization, a number of factors like the pH, humidity, sweat levels of the skin, as well as presence of other bacteria on our skin, all affect the amount of Staph aureus that's present. If more and more Staph aureus is around on the skin, it starts to penetrate through tiny microfissures in the skin, like you get with eczema, as well as larger breaks in the skin like you might get after shaving. In fact, it's particularly troublesome in terms of causing wound infections, where there's a large break in the skin either from trauma or after a surgery. So low levels of Staph aureus with intact skin leads to colonization, whereas high levels of Staph aureus with breaks in the skin leads to infections. When Staph aureus invades into the skin, it can lead to localized skin infections, like a pimple, which can evolve into a furuncle, or a boil. A bunch of furuncles clustered together make a carbuncle. There can also be diffuse skin infections, like superficial impetigo, which is an infection of the epidermis, or deeper-reaching cellulitis, which is an infection of the dermis and can spread over larger surfaces rapidly. If the infection goes deeper, it can develop into a subcutaneous abscess, which is a collection of pus that's walled off and sometimes develops thin walls within it, called septations. These abscesses can happen all over the body, including in the mouth, where they're called dental abscesses, and they can develop within various organs like the liver, kidney, spleen, and brain. Now, if the infection is overlying a muscle, it can spread into the muscle, causing pyomyositis. If it gets into the bone, it can cause osteomyelitis. And if it gets into the joint space, it can cause septic arthritis. Finally, if Staph aureus gets into the bloodstream, it can cause a septic thrombophlebitis, an infected blood clot. In addition, bacteria in the blood is called bacteremia, and it can lead to a number of serious problems. There's typically a widespread immune reaction that causes the blood vessels to expand, and the blood pressure to fall. The result is hypotension and poor perfusion to various organs, a process called sepsis. Once it's in the blood, Staph aureus can also get to various parts of the body. It can get into the central nervous system, causing bacterial meningitis, or an epidural abscess in the spine. It can get into the lungs, causing severe pneumonia. It can start to grow on the heart valves in clumps called vegetations, damaging the valves, a process called infective endocarditis. Bits of the vegetations can then chip off and embolize further, causing other local infections around the body. Now, in addition to invading the body through the skin, Staph aureus can also enter directly into the bloodstream when a person is getting surgery or having dental work done. These events happen infrequently, but when they do come up, it's important to take precautions. For example, individuals at high risk of getting serious disease with Staph aureus, like immunocompromised individuals or those that are at risk for infective endocarditis, should be given antibiotic prophylaxis. Another property of our golden staph is its ability to create biofilm on medical implants like indwelling intravenous catheters, prosthetic heart valves, and artificial joints. The biofilm is essentially a layer of slime within which the Staph aureus live. It forms when a cluster of Staph aureus adheres to a surface, either a natural one like the surface of a valve or an artificial one like the surface of a catheter. The bacteria start to produce extracellular matrix made of exopolysaccharides, or EPS, and over time the cells get completely surrounded by it. The cells that are surrounded by the gel-like layer of exopolysaccharides can communicate with one another through biochemical signals, and can even swap genetic information back and forth, including antibiotic resistance genes. In addition, Staph aureus thrives but doesn't divide rapidly within these biofilms, and it's relatively hard for antibiotics to penetrate into the biofilms. Combined, that makes it a lot harder to get rid of these biofilm infections, and often requires simply removing the surface that they're growing on, if possible. If all this wasn't enough, Staph aureus can also release superantigens or toxins. In fact, there are 5 major toxins related to Staph aureus: toxic shock syndrome toxin 1, or TSST1, Pantin-Valentine leukocidin toxin, hemolysin, exfoliatin, and enterotoxin. TSST1 is produced at the site of infection and can enter the bloodstream. TSST1 binds to major histocompatibility complex type 2, or MHC2, which is a receptor found on immune cells called antigen-presenting cells. When TSST1 binds to the MHC class II receptors on these cells, it really stimulates them, making them release loads of pro-inflammatory chemicals called cytokines, creating a cytokine storm. The cytokine storm results in a number of physiologic changes, like fevers, a sunburn-like rash, low blood pressure, and poor end organ perfusion that can result in death. And together this is called toxic shock syndrome. Pantin-Valentine leukocidin toxin, or PVL, punches holes in leukocytes, our immune cells, which kills them, and gives the toxin its name. Leukocytes with holes in their cell membrane die through necrosis, and that triggers inflammation. When that happens throughout the tissue, like in a necrotizing pneumonia, large chunks of the tissue starts to die off, and the organ can't do its job properly, and this leads to organ failure. Hemolysin is another pore-forming toxin that destroys erythrocytes, or red blood cells, releasing their hemoglobin, which contains iron, into the blood. Staph aureus uses iron in its own metabolism, so this is one way it gets access to that coveted heavy metal. Exfoliatin toxin causes staphylococcal scalded skin syndrome, or SSSS, or Ritter's disease. It creates painful patches of red skin, with fluid-filled blisters, but it often resolves within a few weeks. Finally, there's enterotoxin. Staph aureus might land on food and start to generate enterotoxin. The enterotoxin is quite stable in the environment, and can remain active even after the bacteria are killed off by cooking. The toxin can even withstand boiling at 100 degrees Celsius for a few minutes. If the toxin is eaten, it can cause food poisoning, with symptoms like vomiting and diarrhea a few hours after ingestion. And in rare situations, if the enterotoxin somehow gets into the bloodstream, it can cause toxic shock syndrome, much like TSST1. The main treatment for infections is antibiotics, but Staph aureus adapts quickly and has developed resistance to a number of antibiotics. Penicillins, which are beta-lactam antibiotics, were tried early on. They work by disrupting the enzyme DD transpeptidase, which is a penicillin binding protein, or PBP. PBP is in charge of cross-linking the long strands of aminopolysaccharides that make the peptidoglycan cell wall, running in parallel. These are made of segments of N-acetylglucosamine, or NAG, and N-acetylmuramic acid, or NAM, in an alternating pattern, so NAG, NAM, NAG, NAM, and so on, like a pearl necklace. Protruding from the tips of the NAM subunits are tetrapeptide and pentapeptide chains. These peptide chains can link to other peptide chains from the neighboring strands through a process known as transpeptidation. If PBPs aren't allowed to work, no peptidoglycan is made, and the cell wall becomes leaky. So when the bacteria try to divide and make more cell wall, they simply can't, and they die. Nowadays, though, practically all strains of Staph aureus make beta-lactamases, which allow them to simply disable beta-lactam antibiotics by breaking down their beta-lactam ring. Next, antibiotics called beta-lactamase inhibitors, like clavulanic acid, were used to bind and disable the beta-lactamases. In addition, new types of beta-lactam antibiotics, like methicillin, were identified that couldn't be easily destroyed by the beta-lactamases. Unfortunately, some Staph aureus strains found a way around this as well, by evolving and expressing a gene called MEK-A, which encodes special PBPs that are unaffected by beta-lactam antibiotics. Methicillin and older penicillins can't physically fit into them, rendering them ineffective. These methicillin-resistant Staph aureus strains are called MRSA for short, and have become more common around the world. There are two major categories of MRSA: the healthcare-associated MRSA, or HA-MRSA, and community-associated MRSA, or CA-MRSA. HA-MRSA is found in places like hospitals and nursing homes, where there are lots of chronically ill patients with a high usage of antibiotics. CA-MRSA is found in the community, and is thought to be due to rampant antibiotic use on factory farms, and overprescription of antibiotics. Both HA-MRSA and CA-MRSA pose a major problem, because infections with those strains cannot be treated with beta-lactam antibiotics. As a result, another class of antibiotics, glycopeptide antibiotics, like vancomycin, are often used instead. Unfortunately, it's not as effective and comes with problematic side effects. To make matters worse, some Staph aureus strains have developed intermediate resistance to vancomycin, and they're called vancomycin intermediate Staph aureus, or VISA for short, which is not the type of VISA anyone wants to get. The strains with complete resistance are called vancomycin resistant Staph aureus, or VRSA. The search is on for new antibiotics and vaccines to prevent Staph aureus infections, but the overuse of antibiotics makes rapid resistance an ongoing problem. Ultimately, treatment of Staph aureus infections involves testing the bacteria against a handful of antibiotics and then choosing the most appropriate antibiotic from them. Sometimes hospitals put together an antibiogram, which shows how frequently bacteria are resistant to a particular antibiotic. Typically, when treating MRSA, antibiotics like clindamycin or vancomycin are chosen, but alternatives like tetracyclines, trimethoprim or sulfamethoxazole, linezolid, tigecycline, daptomycin, and quinopristin dalfopristin. Alright, as a quick recap, Staph aureus is a gram-positive coccus that grows in clusters. It's a part of the normal skin and nasal flora in about a quarter of the population. But if it overgrows or if the skin is damaged, then it can cause disease through direct colonization, toxin production, or both. Staph aureus is very adaptive, and it's become resistant to various antibiotics. Methicillin-resistant strains are often classified as HA-MRSA or CA-MRSA, and vancomycin intermediate and vancomycin-resistant strains are called VISA and VRSA.